Markers are used to identify things. Okay, and just for example, you had earlier, uh, if you look at, for example, say some crop varieties, and you see that one has got the disease, one does not have the disease. So then you say, okay, this is resistant, the one that does not have a disease, and the one that has the disease is susceptible to that particular disease, right? So markers can be of different types. Yeah, for example, when you go to ईदगाह गए हो कभी तुम लोग बकरा लाने? Or do you see like these sheep, goats, they have different colors on their backs, pink color, blue color, red color, green color.
Yes, sir.
That-
Yes, sir.
Yes, yes, yes. Why is that?
स- पहचानने के लिए कौन किसका है?
Maybe phone kiska hai because there are so many vendors there. Yeah, for example, they want to distinguish between the one that's costly and the one that is not that costly, the one that's young. So to distinguish, right? So for example, if I tell you now you have a rice variety that you want to know it's resistant, and another variety is susceptible, you want to know it's susceptible. The thing is that you have to wait. You have to sow the crop, sow the seeds, then you have to water the crop, then it will grow, and then you infect them. They'll see, okay, these are resistant, these are susceptible. So it will take a long time. So how using biotechnology, we can determine resistance like this. Even the seeds can tell us will they be resistant or susceptible, right? Or any character for that matter. Okay, if the seed is high yielding or low yielding. Okay, so earlier we used to have a phenotypic marker. Okay, like for example, a tree is tall or dwarf. If you want to see at a phenotype and know कि ये tall है dwarf है, so that's the marker. The phenotype is a marker. Or like we just said, a plant is resistant or it is susceptible. So morphologically we can tell, right? So that these are morphological or phenotypic markers. Okay, so then scientists thought that now this is not going to work with biotechnology because you need things fast, right? So they derived biochemical markers. They derived the concept of biochemical markers, okay, that will tell us a particular protein, say for example, is expressed or not. For example, a plant produces some, say, disease-resistant protein. So what you do, you check the plant and see that they have a protein profile. So we have not done SDS page. We were planning to do a practical today, but since your holiday started, so we could not. So if you guys are interested in having practicals, I don't know, you can come to the lab. For practicals, you can come. So it's your choice. The ball is in your court. If you want to, we can do it. We have no issues. So for example, you've got a plant that is disease resistant. You want to check it biochemically. So you extract the proteins, and then you check the protein on the cell. So like I said last time, for DNA we use agarose electrophoresis. For protein we use SDS phage electrophoresis. And then you have a protein of interest. It will tell us that okay, this resistant protein is present or not. Or for example, you have isozymes, or you run a native page. So, but these biochemical markers were not much successful. Okay, there were not many, and for example, there is a disease resistance that is due to many proteins or many genes. So now to track all those genes is very difficult in these biochemical markers. So it's very difficult and are very unstable. So then came the concept of your DNA-based markers. You can see my board, right? So then we have the concept of genetic markers. And as I said, at seed level also, you can tell that this plant will be having high yield, it will be resistant, it will be susceptible. You'll be able to tell at genetic level. And there are many types of genetic markers, like you have RAPD, it stands for randomly amplified polymorphic DNA. Okay, this is also a DNA based marker. So then you have RFLP, this is restriction fragment length polymorphism. समझ आ रहा है? बताते रहो यार, आप लोग खामोश मत रहो.
यस सर, आ रहा है साउंड.
बस नहीं आ रहा है. Then you have AFLP, amplified fragment length polymorphism. Length polymorphism. Okay, and besides these, you have your SSRs, simple sequence repeats, ISSR, intersequence specific repeats. Okay, so these are main markers we use to differentiate between two organisms, two varieties, right? कोई शक?
No sir.
So what actually these markers do, they check if there is an insertion in DNA, there's a deletion, there is an inversion. So many types of mutations that occur in DNA. Mutations can have many causes. Mutations can be caused by chemicals. Mutations can be caused by radiation. Mutation can be caused when there is homologous recombination, yeah, non-homologous recombination. Mutation can be caused when there are double-stranded breaks. Mutations can be caused if, for example, you have so much damage in the cell that there's a repair we call as SOS repair, SOS repair. So the cell has two choices: either it dies, it has to die through programmed cell death, or it has to mutate itself. Okay, so it prefers to live, and so there's this SOS repair that repairs the DNA in a very random fashion, and then it causes a lot of more mutations. So mutation can have many causes, and these mutations then are detected. These mutations cause variations, and these mutations we detect through DNA markers. कोई बात समझ में नहीं आई यहाँ तक? These are the main markers. इसके अलावा भी बहुत सारे हैं, तो but let's kind of keep specific. So you have, for example, PCR you have studied, right? PCR you have studied, right?
Yes, sir.
Sir,
Is a dominant marker. Okay, there are two types of markers, dominant and codominant. I'll tell you the difference. So this is a dominant marker and this is very easy and fast. Okay. And here, if we, what we do, for example, this is your one DNA, okay, and this is your another DNA, okay. Say type 1 and type 2, any, not type 1, type 2 restriction, don't go there. So now, what we do, we use random primers. We use random primers, and if there are some mutations in the DNA, these primers will not bind. They'll bind to one type, they will not bind to other type. So because these primers are specific, and you know, if you have studied PCR, what primers are, right? So, for example, I use some random primers, so they will bind here, and they'll bind here, they'll bind here, and they'll bind here. They'll bind here, they'll bind here, and they'll bind say here. And यहाँ पर ऐसी म्यूटेशन हो गई इसमें कि यहाँ पर ये बाइंड नहीं हुआ. Okay, because primers are nothing but short oligonucleotides that are complementary to a DNA and that bind. And if they are perfectly complementary, they easily bind. If they are not complementary, they usually don't bind. Now you know that two primers are needed for PCR to occur. So when we run a PCR with these primers, you will have a band, okay, that will be amplified here, and a band that will be amplified here, right? इस केस में एक बैंड यहाँ पर एम्पलीफाई होगा, मगर इसका रिवर्स प्राइमर नहीं है, तो ये एम्पलीफाई नहीं होगा. सो व्हेन यू रन अ जेल, इसमें हमें दो बैंड मिलेंगे, ओके, और इसमें हमें दूसरे वाले टाइप में एक ही बैंड मिलेगा, जिस बेसिस पर हम इनको बाइंडिंग पैटर्न की बेसिस पर डिफरेंशिएट करते हैं. तो डीएनए मार्कर नथिंग बट दे डिफरेंशिएट our DNA of 2 types or 3 types or 100 types or 500 types with its bands, presence or absence of band. Okay, like you have, then how do you read this band? Like you have ones and zeros in binomial, right? Binomials, binomial बोलते हैं इनको, ones and zeros, binary digits. So like computer language, that's ones and zeros. So if there's a band, we say it's 1. Here's a band, we say 1. Here's a band, we say 1. Here's not a band, we say 0. So ones and zeros will tell us that the binding patterns, how they are, and then we can kind of use computer programs to read these binding patterns. So, but क्या principle है? Simple है. If there's a mutation here, this reverse primer will not bind at any place, and when you run a PCR, these will get amplified, but this will not get amplified. So there will be a difference between these two organisms. This has 2 bands, this has 1 band. कोई शक?
नो सर.
अब कभी क्या होता है कि अगर बैंडिंग पैटर्न में हमने ये अब देखा, इसमें ये एक्स्ट्रा बैंड है, इसमें नहीं है, लाइक ये कॉमन बैंड है, ठीक है, दिस बैंड इज ओनली प्रेजेंट इन टाइप 1 एंड टाइप 2 में नहीं है. अगर हम इस बैंड को यहां से निकालेंगे, जेल प्यूरिफाई करेंगे एंड फिर इसका सीक्वेंस करेंगे, इस मार्कर को हम बोलेंगे स्कार. these are your scar markers. You sequence it and know कि ये sequence किस जीन की है, क्या है, कैसी है. Okay. For example, if you have a difference in your binding pattern, there's a band that's monomorphic. Okay, this is a polymorphic that is present in each and every. If there's a band difference in binding pattern, you take this band that's extra, present in 1 and not present in 2. Okay, आवाज आ रही है? So you will excise this band, okay, get a sequence, and then the marker, then you get the sequence. So this is as a scar. We use call it a scar, sequence characterized amplified region marker, or a scar marker. So RAPD can be of 2 types, AP-PCR and DAF. AP-PCR and DAF are 2 AP, PCR, and DAF. These are two RAPD base markers. One is the arbitrary primed PCR, in which we use long primers. And in DAF, we use short primers. एंड इसमें जो हमें एम्प्लीकॉन्स मिलते हैं वो बहुत सारे मिलते हैं. इसमें एम्प्लीकॉन्स हमें कम मिलते हैं. एम्प्लीकॉन्स मीन्स अगर फॉर इनका हम दोनों देखें तो क्योंकि ये लॉन्ग प्राइमर्स होते हैं, ओके, इनमें एम्प्लीकॉन साइज हमें देर मोर स्पेसिफिक, एक या दो मिलेंगे. डैश में हमें क्योंकि शॉर्ट एम्प्लीकॉन्स होते हैं, सो दे विल हैव शॉर्टर, they will form more amplicons. After that, you have your SSRs. Simple sequence repeats. आवाज आ रही है बच्चों?
आ रही है.
So SSRs are your simple sequence repeats. Okay. They can be of three types: STRs, microsatellites, and SSLPs, simple sequence length polymorphism.
Sir, repeat, sir.
SSRs can be of three types: STRs, microsatellites, as well as SSLPs. STRs are short tandem repeats, microsatellites, and SSLPs are your simple sequence length polymorphism. So the repeats that here are, say for example, mono repeats. Mono repeats means A 10 बार, 20 बार, 30 बार, 40 बार, जितनी बार भी हो, ठीक है, but should be greater than 12, greater or equal to 12. Then you have die repeats, जैसे 80 है, 80 कितनी बार, 80 80 80 80 80 जितनी बार भी आप इसको रखें, तो शुड बी ग्रेटर देन 6, तभी इसको हम डाई रिपीट कहेंगे. ग्रेटर इक्वल से. इसी तरह आपका ट्राई रिपीट्स है, ठीक है, टेट्रा रिपीट्स है, पेंटा रिपीट्स है, जैसे फॉर एग्जाम्पल ATGCT, फिर से हो गया ATGCT, फिर से ATGCT. So this ATGCT 1 2 3 4 5 is a penta repeat that is repeated after, it should be more than 5, okay, or equal to 5. Now these SSRs can be genic or genomic. Can be, are of two types. Genic or जीनोमिक. जेनिक मीन्स जब ये किसी जीन के पार्ट से हम कोई मार्कर बनाते हैं, देन इट विल बी कॉल्ड एज जेनिक. These markers are in your coding regions. You know that DNA has introns and exons, and introns are does not code for a gene. So जो exons होते हैं, ये usually exons होते हैं जिनसे मार्कर बनाए जाते हैं, ओके. एंड जो जीनोमिक होते हैं वो नॉन कोडिंग रीजन से बनाए जाते हैं. नॉन कोडिंग मीन्स they do not form a part, or they are not expressed as RNAs or proteins. Okay. These show less variability. Sorry. These are more variable and these are less variable. ठीक है, so these are SSR your codominant markers. That's why you prefer it over dominant markers, क्योंकि ये हमें heterozygous condition भी बताता है. So if you want to design primers for SSR, there are various tools. There's a tool called as NISA, online tool है, और SSR IT. These are tools online. आपको सीक्वेंस देंगे या आपको SSR प्राइमर्स बनाकर दे देगी. आप कभी क्या, जैसे फॉर एग्जांपल ये सीक्वेंस है, जैसे ये सीक्वेंस आपकी ATG ट्राई न्यूक्लियोटाइड रिपीट है, ओके, एंड यहां पर भी आपको ATG है. So these sequences will have an inter region. ओके, हम क्या करते हैं, हम प्राइमर्स इसके और इसके स्पेसिफिक बनाते हैं, जिसकी वजह से हमें ये इंटर रीजन है, ये एम्पलीफाई होता है. दैट्स व्हाई इसको हम बोलते हैं ISSR, इंटर सिंपल सीक्वेंस रिपीट्स, और ISSR, दीज आर द रिपीट्स. SSR में आपने देखा कि हम इसको एम्पलीफाई करते हैं या इसको एम्पलीफाई करते हैं, मगर इनके बीच में एक रीजन होता है जो वेरिएबल होता है, उसको भी हम एम्पलीफाई कर सकते हैं, जिसको हम ISSR बोलते हैं. ठीक है, कोई शक?